do these 4 half sheet questions. So go ahead and take a few minutes to do that now. that you struggle with those for a while, I realize that the female reprophysiology is packed. And I don't know how much you've been studying, but these should have been taken straight from your notes. So I'll give a little assistance. For example, for number 1, do you understand about the completion of Meiosis 1? If you were referring to your notes, I kind of drew like a picture like that. And they said you ejected something, and the cell kept everything. And I can't remember the chromosomal pattern I did, but I always do one long, one short. That's when we kind of talked about this. This would be the completion of Meiosis 1. What's the name of this cell? Which one? First or second? First. What's the name of this cell after you complete Meiosis 1? It becomes the secondary oocyte. I drew 2 of these. What are the X's called? Dyads. So how'd you answer number 1? What's here and what's here? 23 of these, okay? 23 dyads. Now, in which stage of follicular development does the primary become the secondary? So, I said 1 achieves dominance, okay? So it's the 1 secondary that becomes tertiary. So there's no strict answer for number 2, this first part of the question. It's the 1 that achieves dominance. So that stage of follicular development, it could be late secondary, early graphium. This is an easy question, I think. Ovulate into what? Uterine tube, yeah. Okay, this is straight up meiosis. And now in phase 1, you're separating what? Yeah. You pull these apart, you pull these apart. Always think of them as sets. The green set belongs to maybe mom, and the purple set belongs to dad. So you're separating the homologous chromosomes in anaphase 1. That's very important to understand. Anaphase 1, you just pull it apart. This is why this is absolutely important to understand. You got a set from your mom, a set from your dad. That's your inheritance. When you pull these apart, you lose half a set. One cell is going to end up with half a set. So what do we call that? Haploid. This doesn't happen in regular cell division. In regular cell division, you line it up like this. Long. These 2 long ones are homologs. And then short would be you line it up single file, and then you pull each one apart in regular cell division, not meiosis. Okay, let's say you pull it apart like this. Here's 2 chromosomes. They're copies of each other. Each one, it's a copy. You're not separating the homologous chromosomes. This is a homolog to that. And as you pull it apart, each cell is getting the same homolog, okay? So you're not separating homologous chromosomes here. As you pull it apart this way, this cell is going to get that chromosome, and it's getting the homologous chromosome with it. That's regular cell division. That's why in meiosis, this first division is very important to understand. That's why it becomes haploid, okay? However, it's not just haploid, it's haploid, but it's still replicated. That's why you need the second round. So I think that's a point, as I check your labs off, you're not quite getting that yet. After the first division, it becomes haploid but replicated. So, in round 2, what do you pull apart? Yeah, you pull apart the sister chromatids, okay? So that way, each cell will get It's still half, it's just not replicated anymore. For number 4, in the female, what had to be present? Sperm had to be present, okay? Well, obviously, I mean, wouldn't that be after ovulation? Sperm ain't gonna be there if you don't ovulate because it's stuck in the ovary. Okay, unless your sperm can swim all the way inside the ovary, which I haven't heard of. Okay, well, if you struggle to answer those, good, because you learn from struggling through the material, and eventually you'll master the material and it won't be a struggle. Then I gave this lecture, which is your essay portion of the test. And I didn't show this slide yet. Maybe let's struggle a little bit more. So we're on 5, 6, 7 now. For 5, 6, and 7, what I want you to do, name the 3 feedbacks that we talked about in that lecture, from ovary to hypothalamus and pit, for 5, 6, and 7. They're shown here. Just describe them. Give you a little less time. All right, this one, I'm not gonna like describe the answer, but I'll point you in the right direction. I really want to see what you put. But I will point you, this is like the first feedback, it's a negative one. Do you know what it is? Can you describe that? The second one is a positive feedback. It's right there. But what's causing it? Well, what's the reason? And then the third one is also a powerful feedback, but it's negative, and it came from here. So I want to see if you can describe this negative, positive, negative feedback. I look for them on half sheets. I'll look for them on your essays. That figure talks about it, and we did discuss it in your notes. For example, it's shown on this figure in nice pictures. When I talked from this, I said that the first feedback, you can see it right here, the negative feedback, turning that off. And I said the second one's a positive feedback. Oh, it's causing this thing to surge. Okay? And the last one is a negative feedback. It made the surge go down. Okay? So I talked about it there in your notes. And hopefully if you're studying the material and oh yeah, okay, it's starting to make sense. The next slide, to bring it back to the PowerPoints, I took pictures of a board that we have that shows female repro for physiology. And let's see if it's here somewhere. A menstrual cycle chart. So we pull them out. We got two of them. Anyways, that's where I took the pictures from. And it's something to look at the review. For example, in the middle, it's hard to see, it's on a blue background. That's day 28. And this is like going through all the way. Okay, that's day 14, they show ovulation there. They show the different phases of the ovarian cycle. But what I also like is they show a picture of the uterus in different cycles too. And they give you the days of the month there, 28 to 5. What day is that? Menses, and that's supposed to be the shedding of the endometrial lining. And then they show the other phases as well. And you can identify it based upon just the days that they give you. 5 to 13, I said 6 to 13, okay, that's fine. But it's the proliferative phase, and you can also tell it's a proliferative phase, not so much by looking at their illustration of the endometrium, but what's going on here. You see larger, more mature follicles. And that's usually what happens. It's just before ovulation. And then they give 13 to 15. Now they showed implantation, but it wouldn't occur until about 7 to 10 days later after fertilization, which would happen on this day. But you know, it's a board, so they have to show everything one at a time. And they put both of these things on one shot: fertilization, implantation. Then they show, well, I think you get the point. That's the secretory phase. And I have some more questions here, but I think we've had enough half sheet questions. But these are, I like to ask a lot of questions the day after that lecture. And it's a wake-up call. If those 7 questions, you're like kind of lost, you kind of know where you are in your studying, don't you? If you're on top of it, boom, oh yeah, I got it. Or maybe you don't got it, but you understood everything, and it's just a matter of referring to your notes. But you're somewhere between, I'm completely lost, and I totally get this. And figure out where you are, and figure out where you need to be, and it's on you, right? I'm doing male repro physiology today. So think about repro is got to do everything twice because we have 2 biological sexes. It's different in both. Some things are the same. For the physiology, I spend a lot of time talking about the spermatogenesis, the formation of sperm in males. Male, repro, spermatogenesis, just like the name suggests, genesis, formation of what? Sperm. So it's outlined right there. Mitosis, meiosis, spermiogenesis. That's the process. Mitosis, meiosis, spermiogenesis. The first two in red, those happen in the female, in oogenesis. There's mitosis. It happens before birth. Then I said it twice now, at the 7th month of pregnancy, follicles form, and it stops. Not for males. As long as you have mitosis of the cells that become sperm, a male can be fertile all the way into their 70s. Okay? But in females, it stops when you get that follicle development. There's menopause that happens. There's no menopause in males. Mitosis keeps going, you always have sperm. But you gotta take some of those cells and you have to make them go through meiosis so they have half the genetic material, right? And when you fertilize the oocyte, you restore the full complement of the human genome. Females did not have this. That secondary oocyte, there's no morphological changes because it doesn't have to travel very far when it's ovulated. It gets swept up into the uterine tube and that's it. Sperm have a long journey, so they have to change their shape to become swim ready. So that's a process that I'll describe. Now, it happens in seminiferous tubule. Here's a picture of it. So what you're looking at here, we looked at seminiferous tubule under the microscope. If I drew just one little cut tube, lumen, that's where sperms swim away. All the development happens, this tube that has a simple columnar epithelium. I'll just draw it on half there, simple columnar epithelium. Seminiferous tubule. Now usually these simple columnar cells, you can't see the full cells because they're covered with the developing sperm. We call them sustentacular cells. Sustentacular cells. An older term that I still use is the Sertoli cell. Okay, you may see either on your exam. The thing about these cells, they have these tight junctions that kind of staple them together. Tight junctions. If I drew these tight junctions all the way around, tight junctions. Okay, all these little things here. It creates these compartments. And so now I'm going to get into a lot of terms. And before I do, the terms are kind of where things get confusing. This class there's been a little more chatter than I'd like to hear. And it can be a distraction to the people around you. So I would ask that you keep the chatter, just cut it out. So when I go through terms, people get kind of confused. People's talking in the background. So please cooperate with me on that. I think that's what's difficult about this lecture, or any lecture, all the terms, but this one can get a little bit out of hand. But anyways, the reason why I present the tight junctions create these boundaries from here to here. and from here to here. We call this compartment right next to the lumen the adluminal compartment. Okay. Put a little star next to it. This part here, the outer part, is called the basal compartment. So the tight junctions create those two compartments, is what you need to understand. In the basal compartment, you have this cell type. Let's go to the top of that figure there. Basal compartment out here, adluminal compartment here. This cell here is called a spermatogonium. It's undergoing mitosis. This is a cell that will become sperm. But right now, it's just undergoing that first part of spermatogenesis, mitosis. Regular cell division. So if I draw one cell up here, they say 2N, diploid, right? One cell becomes 2, so it divides. In regular cell division is just one cell becomes 2, and the genetic material is the same. 23 pairs of homologous chromosomes in each. Okay, and we designated A and B. Okay, that's shown on the slide here. The A's remain in the basal compartment, but the B's are pushed to the adluminal compartment. So if you're I'll just put a circle here with the A in it. You just stay there, and you continue mitosis, right? You have to stay here so the man can remain fertile. As long as you got mitosis of these cells, you're always going to have sperm. You won't run out. But you got to take some of them to become sperm. So you take half of them, say the B's, this one, and you push them to the adluminal compartment for further maturation. You push them past those, they squeeze past the tight junctions of the sustentacular cells. So that's what's going on there. So you take that cell, you push it. Now it's going to enter the meiotic process right here. You call a cell now a primary spermatocyte. 2NR. They put 2N, I put 2NR because we know what that means. It's haploid, it's not haploid, it's diploid replicated. And you have the 23 tetrads, that whole thing. So this cell is a primary spermatocyte. So this is the same conversation we've been having, right? So for example, if I draw the primary spermatocyte here, with my usual 2 chromosomes, or 2 tetrads, instead of 23, that's 2NR, right? And I'm just going to draw it just like that, because what you have to understand, this primary spermatocyte is going to divide into 2 cells. way I've drawn the metaphase. What are you going to put in this nucleus? Long green, short green? That's kind of how I'm leading you, right? So we'll process as you go. That's how you draw. This is meiosis 1 being completed. So I'm just kind of following this figure here. Now you call these two cells secondary spermatocytes. So I just did meiosis 1 in one shot. You started off with a primary spermatocyte. With 23 tetrads in the cell. Then you complete meiosis 1, and you have 2 secondary spermatocytes. Secondary. I drew 2 dyads in each cell, but really it's 23 dyads. And each of these cells is haploid replicating. The cell it came from, the cell is 2NR. Okay, so the whole 2NR, NR, hopefully you're following that, okay? Now the reason why I drew like a dumbbell is the cytoplasm connects them. These cells, they need each other. They have to share the cytoplasm. They work better together. They survive better by having this unitary cytoplasm. So they stay connected, these two cells. One cell becomes 2 in meiosis 1. 2 cells become 4 in meiosis 2. So that's what they show there. So let me just go, 4 dumbbells there. Okay, so this cell is going to divide into that cell and that cell. The question becomes, what do you put in each cell? Well, put one long, one short green. Long green, short green, long green, short green. And then we're going to put in these two cells. Long purple, short purple, long purple, short purple. Okay. So in each of these cells, what you call them spermatids. You don't call them sperm yet because they're not swim ready. But what's in each of those 4 cells? 23 single chromosomes. We're just haploid now. Not replicated. Haploid, 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 haploid. Okay, well, let's see. 23 single chromosomes. So to recap all the names of the cells, spermatogonium, type A, type B. The B's become primary spermatocyte, then you go secondary spermatocyte, then spermatid. So keep the names of the cells straight in your head. Keep the chromosomal pattern of each cell. What's in this cell? What's in that cell? What's in that cell? Okay, any questions yet? This is meiosis. Okay?
All primary spermatocytes are in the abdominal compartment? Is that what
Yeah. They're pushed, the ones that enter meiosis are pushed to the luminal compartment. That is correct. So check the box. We did step 1, mitosis of spermatogenesis. Step 2 is all this, meiosis. Check that. with step 3, these have to become swim ready. So they use early and late spermatid. I usually don't use that terminology, but the process of becoming swim ready is spermiogenesis. Okay, let's talk about that. This whole process, mitosis, meiosis, spermatogenesis, 70 days. 70 days to make little sperm. Spermatogenesis is the last 24 days of that 70 days of this process. Okay? You're done dividing the genetic material. It's basically just spermatids are undergoing morphological changes, change the shape, to become swim ready. That process is shown, well, you can't really see it here, but I did want to show you a real seminiferous tubule. It is possible to identify some of these cell types, although I won't make you do that. Okay. I think it's enough just to know the names and some kind of easy chart like that one. Okay. Students will struggle with that enough. That's good enough knowledge for us, I think. All right. Well, anyways, this shows in about 24 days, process of becoming swim ready. So look at the left, that's kind of where you start with these cells. Regular round spermatid, right? I'm going to erase this now. So what do we got there? Regular round spermatid. Got purple nuke. The one thing I'll draw is Golgi. Golgi apparatus, stack of flat pancakes. That's what it looks like under the microscope. Okay. Now the nucleus is just haploid DNA, right? Now look what happens. The Golgi, it forms this cap around the nuke. And you're dropping out the cytoplasm and you're getting a mid-piece tail along the way. So that's the one thing I'll emphasize. So that Golgi becomes more around the nuke. Here's a nucleus, little rocket shaped thing. It has a double membrane. We call that the acrosomal cap, or just the acrosome. Acrosomal helmet surrounding the nuke. With I'll just put N for haploid DNA, and around that you have cell membrane. And then you have mid-piece tail. So the head is the important part. It's got that acrosomal helmet. That helmet is important. It's not just a helmet for helmet's sake. It's got all these little dissolving chemicals, lytic enzymes as they're called. You want to keep your acrosomal helmet on. You get close to the egg, you want that to spill and dissolve holes to get to the egg. Okay, we'll talk about that later. But for now, got your little helmet. Let's look at another picture. Sperm structure. we've been calling this sperm. The full name is spermatocyte. Here's the structures there. Head, which I've said. The neck would be right here. The neck can pivot. It's important that it can pivot because you have a rocket-shaped head. When it wants to fuse with the membrane of the oocyte, you want it to like turn so it has more surface area that can like fuse with the oocyte membrane. So you want that neck to pivot. You got a mid-piece. Here's my mid-piece right there. It's got mitochondria. Energy for whipping the tail. For locomotion. A fertilizing sperm, if it's the winner, the tail will help it like corkscrew a little bit, so it kind of drill its way inside there. Okay. Here's a real histology picture. You can see cells at 400x under the microscope, but look how much you got to go in to get this view. Sperm cells are very small. This is 17,000x. What do you think AC stands for? Well, it's acrosomal cap. I call it helmet. PM is plasma membrane. You can see all the mitochondria in that mid-piece. You can see down the central axis, you have a nice structure for a tail that can whip. Okay, so that's sperm. This figure here, the Sertoli cells. I had already mentioned that they're called sustentacular cells, and I already mentioned the tight junctions. I drew them as simple columnar. Here they just draw the cytoplasm. Pretty much the cytoplasm of these cells, they kind of bathe and protect and kind of nurse the developing sperm. That's kind of their functions. They're like nurse cells. They provide metabolic support. Their cytoplasm develops the spermatocytes. When a sperm's ready to swim, ready to leave its little nest here, it's got its head stuck in the sand, and it needs to break away and swim away. The process of it breaking away, taking its head out of the sand, so to speak, is called spermiation. So we had spermatogenesis, spermiogenesis, spermiation. A lot of derivations of the word sperm. This one describes the process of sperm breaking free from tubule wall. And we know where they go. We went through the whole male duct system. Seminiferous tubule, rete testes, efferent ducteals, epididymis, vas deferens. Went through that whole song and dance, right? We know where sperm can go. But them breaking free is dependent upon testosterone. I abbreviate testosterone, capital T sometimes. Okay. Thing is, something's wrong. You have low testosterone. You don't break free. And maybe the Sertoli cell just gobbles you up. You never get out of there. Okay? The other thing that could be problematic with low T are those tight junctions, those little adhesions. With low testosterone, those adhesions can break down, and sperm cells can slip through too quickly and they never develop. Tight junctions break down. Sperm get through too fast. Don't develop. Okay, so the name of the game is safety in numbers here. We want to make many sperm as possible. Young man in their 20s to make something like 6.5 million sperm per gram of testicular tissue. Get to age 50, divide that number in half. Okay, but just to give you an idea, the goal is make as many as possible. You only need one to fertilize, but you need millions to start off with. The other thing I'll mention that's on this slide is that this tight junctions, they form a blood-testes barriers. Your immune system would see a developing sperm as a non-self cell, so it would attack it. So it's very important to have this barrier to protect the developing sperm from immunoglobulins in the blood. So I'll put that as well. Tight junctions create a blood testes barrier to protect developing sperm from immunoglobulins. We have models of cells undergoing spermatogenesis. A student took this picture in 5, 6, 7 there. Well, anyways, to get you started, they're over there. You can see them. But take a close look. It's hard to see from a picture, but when you have the model right in front of you, you can see that's a double, that's a double. So you're separating the homologous chromosomes, correct? So if I said name the phase, what would you put? Anaphase 1. Very good. And I would ask you the gender too. It's male. How do you know it's male? Because female, isn't it an unequal division? The whole polar body thing? Yeah, that's how you can tell. Gender, biological sex. So if this is anaphase 1, what's this one? Anaphase 2. Looks like you got some swim-ready cells. The name of these cells just spermatocytes, I guess, right? Sperm. But what's the ploidy there? If it's like 2N, is it 2NR? Is it NR? Is it N? Which one is it? Just N. N here, N here, N here, N here. Eight, 9, and 10. What if it's a female? Anaphase 1. Torn apart homologous chromosomes. That's a double, that's a double. These are singles. Anaphase 2. This is the first polar body. Right here, would you agree? Can you see it in this model? The first polar body is right here, undergoing a division. Okay? Now this is the first polar body, what's out. That's the second one. Now, you're supposed to be stuck here until sperm arrive. I guess this assumes sperm is there, because it finished, right? So this is supposed to go to the sperm DNA, which is not shown. That's okay. I got 3 polar bodies, that's the first, that's the second, that's the first. These 2 came from the first, that's the second polar body. But that's what's important, okay? So you should be able to tell male and female. Sometimes I like to put them together. I'll say, what's the phase of, identify phase. These are both what? Anaphase 1. But which one's female? the one on the left. Okay, and the same thing here too, right? I'm going to shift gears from cell division, meiosis, and talk about the hormonal control of spermatogenesis. We got this picture again. Now in males, it's a lot easier to understand because there's no cycle I have to teach. It's just, yeah, there's no cycle.
So if you have low testosterone, does that mean you're sterile?
Maybe just, I don't know. No, not necessarily. You just cycle high, cycle low. There could be a clinical reason why you have low testosterone, but I think a normal person, normal man, there are moments when you have low testosterone. Okay, but there's no textbook reason why, because men don't have a cycle, so I can't really teach that. But what we learn from this figure is it does get high, it does get low, and when it gets low, you have to turn this thing on. So let's kind of look at this. It's the same hormones. Hypothalamus and pituitary. Make a column for that. So you got your whole GnRH, FSH, LH. The gonadotropins from the pit. Now, it turns out these gonadotropins are named for what they do in the female. We just keep the names for the male. It's very interesting. This is the only time I've seen them do that. Let's just keep it simple. Just have one name for both. There's no follicles in men. There's no luteal phase, but we just keep the names. At least that they kept that simple for us. So the thing is, if say you got low testosterone, low sperm count, you want to increase it. Okay, that is kind of the stimulus. For GnRH, when those things are low, I'll say low T. FSH and LH do different things. FSH, it's going to stimulate those sustentacular cells to increase these androgen binding proteins in the area. So those will start to create androgen binding proteins. So they'll kind of like sop up the testosterone that's being secreted by what cell? Interstitial cells of Leydig. Taught you that before. Okay, they'll kind of localize it to the area, and that'll help. Testosterone does help the spermatogenesis process. Okay. They bind testosterone. Testosterone helps spermatogenesis. Okay. Well, LH stimulates the interstitial cells to secrete testosterone. The interstitial cells, they're like the theca in the female follicle. Anyways, these cells secrete testosterone. They secrete the T, and if you got the androgen binding proteins around, the testosterone some of it will enter the bloodstream for male secondary sex characteristics, and some will help the development of sperm. They'll be bound by the androgen binding proteins. So when you have high testosterone, you have increased sperm count. That's inhibition. So what about athletes or anybody, female bodybuilders, for example? They inject testosterone. What's the effect on this axis here? You inhibit. So what happens to the testicles in a man? they shrivel up. Okay? So you get big as a house working out in the gym, but then your testes shrivel. Counterintuitive, if you ask me. What happens to a female bodybuilder? The voice gets deeper. Why? I don't know. There must be androgen receptors in the voice box. That's my thing. Maybe it gets thickened as a result. But anyways, just let biology do it. Don't inject these things. I want to shift gears. So that was pretty simple, right? Males compared to females. It took me like 40 minutes to teach the females. For this, it's just, there's no cycle. It's high or it's low, and turn it on, turn it off. If there's no questions on this, I'm going to move on. Okay, let's do that. Human sexual response. This was first looked at scientifically by Masters and Johnsons. In every book I've ever read, they just base it off their research. And what they observed is excitement plateau orgasm resolution in men and women. Although it is a little bit different in men and women, but they go through those same phases. So in terms of the physiology, I'll just stick to what I have on the slides there. I don't think I need to add any board notes to this. Because we kind of talked about it before. Well, this erection we did, the erectile bodies become engorged with blood. And I described that as a vasodilation event. You also have scrotal swelling. That is an abbreviation, nitric oxide, and that's the second messenger that causes the vasodilation. Let's see. So, erection happens first. The little blue pill. This is some of the research done in the 80s by Louis Ignara. Let's see here. So what he did, he took rabbit corpus cavernosum. Okay. And what he did was, he treated it with nitroglycerin, which was known to be a vasodilator, but the molecule that caused the vasodilation had not been identified. He identified it as the nitric oxide, and he got the same result in terms of the relaxation. And whenever you can identify the molecule that causes the physiology, and you can make a drug that can affect the physiology, then you can help people, right? That's why that was so important. I mean, he won the Nobel Prize. I remember that happened in 1998. It was my first year of grad school at UCLA, and I remember it was a big deal. I don't know, most of you probably don't remember 1998. But do you remember when you started seeing those, if you're old like me, do you remember when you started seeing those commercials for that stuff? It was about that time, early 2000s, they started showing this ED commercials, because this came out and the research was done maybe a decade prior. So anyways, that's the physiology for erection. Okay, it's a vasodilation event, nitric oxide. I mean, everything vasodilates. Nitric oxide, I studied it in heart disease. Heart disease is when your coronary arteries get clogged up, and if you don't have enough nitric oxide, that's a risk factor for heart disease. So it's very important. Not just for the little blue pill. Okay. The male sexual response, when you get to the plateau phase, everything's erect. The fluid you might see at the tip of the penis comes from the bulbourethral gland, clearing out any trace urine in the urethra. In orgasm, the physiology is contraction of penis and internal structures like bulbal spongiosis. There's maybe 3 to 5 strong contractions followed by weaker ones, about a second apart, and you see a spike in cardiology. Remember, in males, orgasm is necessary for fertilization because the ejaculate contains the sperm, and the number I gave was something like 20 to 100 million per mil. Okay, that's kind of what we see in a normal male to be fertile. All right. Resolution, or the refractory period, everything just goes back. The heart rate goes back to normal, everything shrinks, the muscles relax. So in the resolution phase for a male, there's a refractory period. Refractory means a period of insensitivity, where erection is difficult to re-stimulate. This time is shorter in younger men. And if we compare the male sex response to the female sex response, it's more varied. I guess they observed a lot of females, they categorize it as either having one large climax or a couple of climaxes, or no big climax, but a bunch of small ones. But all of those patterns go through this same process of excitement, plateau, orgasm. So in excitement, there's increased lubrication. Now, we kind of understand that. The anatomy that I taught for that is the vestibular, fill in the blank, bulb, okay? That's erectile tissue. In males, it's the bulb of the penis. In females, it's the vestibular bulb. But that's not a gland. But what does that bulb compress? That is a gland. The greater vestibular gland. It compresses the gland. That causes that. Okay. Well, the glans clitoris may swell. There's other things that swell, changes in the labia. The vagina balloons out. So the fornix really becomes enlarged, and the cervix gets out of the way. Okay. So here's some of the changes that they show. Yeah, there's no real physiology that's going on here, other than just to mention these changes. But I think one thing I should mention, during orgasm, it's similar, 3 to 4, maybe up to 12 contractions, about 0.8 seconds apart. It may be repeated, it may follow different patterns. Orgasm is not necessary for fertilization, right? Because for fertilization, what does the female need to do on day 14? Ovulate, but that is not dependent upon the female sex response, okay? I've read that other mammals, ovulation can be triggered with mating, but not humans, okay? Now what they show here is the seminal pool, and that's the fornix, okay? So when you have resolution, the cervix will kind of swoop down in there and basically suck up the ejaculate. That's what's shown here during resolution when everything shrinks back down there. That's important for fertilization. So if it's on the right day, then you have the chance to reproduce. So everything goes back to normal. There's no refractory period in women. Men have a refractory period, women do not. Masters and Johnson report women can be multi-orgasmic up to 5 to 20, and it's not age dependent. So like I say, women have all the fun, right? This is what they report. Men have a refractory period, and it gets longer as we get older. So I guess we have a biological clock too. I want to shift gears to histology. I'll post these slides today. I went through my thumb drive and I found some that are good. And what I'll do is, this is for your quiz on Monday. Wait, yeah, okay. Let me pass out the study guide. I'll just put it out. I think I put it there. A couple of things. Right now, why don't you turn in your half sheet and get one of those study guides if you want to look through it as I present the slides. Go ahead and take a minute.